Okay, so yeah, welcome everyone. I am Klara Kropiuszek-Brumat and I am a SMESH Fellow at the University of Nova Gorica. I'm working at Professor Arjo DeMarco's group and basically at the interface of nanobody discovery, and we're also using machine learning techniques for that purpose. So bioinformatically, but also for structural determination and for helping finding binders. So today we're going to talk about how machine learning cracked the biology's complicated challenge of determining structural predictions. And I hope that the module, oops, sorry. It's a bit stuck. The classical problems of presentations. Okay, yeah. For today's agenda, I decided to start with biology foundations. I don't know, is anyone of you biologist? You are? Oh, my goodness, I did not prepare for this. So I want to be brief about that, and then a bit about the machine learning revolution, from CNNs to transformers, the AlphaFold architecture, then the critical evaluation, where models fail. And here I'm gonna go a little bit into our current research and something we've recently published, at least as a preprint, and then some case study like practical guide, some diagnostic exercise, what you could be maybe using for your work, takeaways. I wrote it's gonna be up till 4 p.m. and then I checked again when I went through it. I think we're gonna be faster and you probably don't need as much introduction as I'm giving, so I'll try to go through it quite briefly. Yeah. Yes. Yes, it all looks good. Okay. So let's start with the biological challenge. What is a protein? Why does shape matter? And how did we solve structures before artificial intelligence? As you know, that biology is digital. The code of life starts with DNA, which is the hard drive, stores instructions, but that is more or less that. I mean, it also has a structure to itself. And then we have RNA, the messenger, which is a temporary copy of the genetic material. And then we go to proteins, the hardware, that are molecular machines that actually do the work. And they can have functional properties, like they carry oxygen, contract muscles, fight viruses, or they can have structural components to it. So they are actually supporting the structure of a cell, of a unit, like integrin, cytoskeleton, keratins, and so on. They can also serve as catalysts to reactions, like enzymes. So there is a lot of actions that they can perform. The key insight is that the proteins start as a simple 1D string of amino acids, and then they must fold into a 3D structure. And the proteins themselves are made of amino acids, as you all probably know. So there is 20 of them. And the proteins are then typically the smallest, from 100 to 1,000 amino acids long. And you can think of a protein as a string of amino acids, when in 1D we have basically just a string of beads, so to say, easily. And then the secondary structure that it takes is a alpha helices or beta sheets that will then in turn help to form a 3D structure of a protein. And a tertiary structure will be when more units are coming together into a functional unit. And one thing that is an important component of a protein is to know that we have, when amino acids are joining together, that happens with a peptide bond. And for that, we need the nitrogen group. Then we have the C alpha atom and the CuO, the acidic chain to the molecule, where the residue can be any one of those 20 amino acids. There are also some artificial and uncommon amino acids that could be, but generally there are 20 amino acids. They can be hydrophobic, charged, bulky, tiny, have aromatic residues to it. And yeah, we can think of it as Lego, where we can just plug and play with them to insert into a certain peptide. Of course, side chains will push, pull, and interact, and that will force a certain chain to fold into a certain a secondary structure, and then later in a 3D structure. Just wanted to illustrate this, that structure somewhat informs the function. So this is heavily related. Shape is kind of everything. If we look at the example of hemoglobin, which you all are familiar with, I'm pretty sure, we see the molecule here on the right, where the heme is colored in red. What is very interesting about this molecule is that upon each binding of oxygen molecule, which we see here, the 3D structure of the whole molecule will change. And then that in turn also changes the affinity for the next molecule of oxygen to bind. So basically, it can bind 4 oxygen molecules. With every conformational change, it becomes easier for the next atom of oxygen to bind. And then, and that happens in the lung, which is a oxygen rich environment. And then when it's going through the body, it's slowly loosening the structure and releasing the oxygen. So a wrong shape, just one mutation can lead to a sickle cell disease, for instance, and that will lead to shape changes and clumps, and the red blood cells might collapse and not function properly anymore. So this is just one example why shape matters a lot, and the conformational changes. And the other case that we will be returning to later on is also antibodies. These are immunoglobulins that have this particular epsilon shape, where we have here the heavy chains, and then we have a bound light chain to it. And with these CDR regions up here, that are called paratopes, they will recognize a certain specific, let's say, viral or bacterial antigen, and they will try to recruit the cells to neutralize a certain beat reaction or stop an infection, for instance. So if you can predict shape from sequence, this gives you a lot of power in understanding the biology, understanding what happens at the cell surface on the inside. But from the basic biology, it goes also to translational, because it can help you maybe to design drugs. And with this, it becomes also a powerful tool in fighting, let's say, diseases. The protein folding problem is also called the Levinthal's paradox. In 1969, the biophysicist Cyrus Levinthal made this calculation that even a small protein has an astronomically large number of possible shapes that it could fold into. And if one computer would be trying to crack all of these possible conformations, it would take like the age of the universe to actually calculate all of the possible states. So this, of course, it doesn't happen in nature. It folds in milliseconds to second, a protein. So there must be a folding funnel. I hope it's okay if I sit. A folding funnel. So we have an energy landscape that guides actually this process. And the question is, can we learn about this funnel from data? Is data enough to learn from it? Not from physics simulation, which was how it was classically often done. And on the other hand, classically, before AI came to help, a single protein structure determination could take an entire career of a person. For instance, one needed to produce a crystal of a protein, and that is not a trivial thing. I know people that did their PhDs trying to produce a crystal and then do NMR or X-ray on it, because one has to fine-tune every step of the process to guide the protein into a 3D crystal state. And for instance, John Kendrew famously spent 12 years for myoglobin. And the funny thing is that he needed massive amounts of pure protein to grow crystals, and horse hearts were not big enough. So he eventually sourced a chunk of whale meat from Peru because, yes, weird, because diving mammals pack their muscles with myoglobin, and whale is an enormously big mammal, so it's a little bit of a, yeah, story from different time. But when the complex finally emerged, it was messy and complex, so very different from what everyone expected. It did win him a Nobel Prize in 1962 in chemistry. So one starts with a crystal, then does, for instance, X-ray diffraction. From that, one learns about phases of the protein. One can create electron density map and then feed atomic model, and then does refinement through several cycles to come to an actual model of a 3D structure. And the process itself can cost a lot. And by 2020, there was around 150,000 structures solved basically experimentally, how people used to do it. And the tools for biological revolution that AI brought came actually from physics. As you all probably know, in 2024, the Nobel Prize in Physics went to John Hupfield and Geoffrey Hinton, that both worked with energy concepts from statistics and statistical mechanics to build networks that store and retrieve information. And Hinton developed a learning algorithm such as backpropagation that is also now fundamental for the majority of neural networks that people use. And you have also heard about it in the lectures before. So physics gave us the tool, but those tools are now really solving some of the deepest biological problems these days. In the very next day, actually, the Nobel Prize was awarded for chemistry, and that went to people who applied those tools then to biology. As you know, Demi Hasabi and John Jumper of Google Mind for AlphaFold, they shared together one half of a prize, and David Baker for computational protein design. So creating proteins that never existed, whereas they determining protein structure from the sequence alone. So for instance, David Baker's team designed TOP7. It's a protein that was entirely different to all known proteins existing in nature with this method. And when they synthesized it in the lab, it folded as they predicted it with their calculations. So that was actually very, it gave a way to the whole new field of de novo design that is right now really kind of like even the hotter topic than the structure prediction. I mean, it goes hand in hand, but it's a very, how to say, also competitive field, because it's like everything machine learning, I think. It's like weekly, in a week you start using a method and next week it's already like forgotten because a new thing emerges. Not so extreme, but this also has a lot of negative consequences that we can discuss later, because nobody ever has time to properly address and benchmark the models. Yes, just some other examples of the proteins that were developed in with Baker's program that is called Rosetta or Rosetta Fold. Maybe you've heard about it, even if you're not biologists. So it enabled them to do new nanomaterials where up to 120 proteins would would link together, and then proteins that bind to an opioid called fentanyl, which is here in purple, that could detect it in environment. Then nanoparticles that can, with influenza virus mimicking, then proteins that function like motors, as you can see here, that have this particular shape and that can power maybe certain reactions in the cells, and also geometrically shaped proteins that can change the shape depending on conditions outside. So this can also be a powerful tool to then do experiments in the lab when you want to test certain condition, or in a case like if you want to produce them for biosensors, for instance. And just some examples of protein structures that were determined by the tool AlphaFold 2. It's like molecular, like this one is around 1,000 proteins that form a pore through the membrane that's surrounding the cell nucleus. So that, for instance, is one of the very difficult molecules to model. The natural enzymes that can decompose plastic, that is also something that the field has been actively working on, because we are drowning in plastic. And also then a bacterial enzyme that causes antibiotic resistance. So these are like some of difficult cases that the field has been able to solve with those tools. And if we want to know how it was possible to come to these structural predictions, we need to go a step back maybe and go from how we came from, or how the field came from CNNs to transformers in the protein structure prediction. Yes? This one? Ah, yes. Okay. Yes. Thank you. Yes. Yeah. Ah, but it's not in my, not on my computer. No, it's blocked. So that's, yes, because I'm in presenter mode. Yes. So yeah, the evolution of machine learning tools for structural prediction, like shortly to give a bit of background to what and why the next thing comes, the transformer. So why CNNs fail for protein, how attention works, and the bridge to biology. I think Suva will go in more detail with transformers and also diffusion not in a later lecture. Yes. So today it's going to be a more soft explanation, not heavy on equations. I hope you, you forgive me that part. Sorry, CNN, yes. CNN is a convolutional neural network, and it is a network that is very good at classifying or learning from images. So when you're looking at the field of an image and sliding through and then putting like different matrices, embedding the images and learning from the patterns that you see on the image. So the standard CNNs that were, or let's say, if we go to the structure solved first in the field, there is this test that maybe you have heard of. It's called CASP competition. It's a critical assessment of structural predictions. It's a 2-yearly blind test where the teams try to predict the structures of experimental proteins that have been solved experimentally but not yet published. And as you can see, the scale is 0 to 100, and this means how closely the prediction is matching the crystal one, the resolved one. And the field was stuck for a long time, around 30 to 40%. And then AlphaFold came and did a big jump, the first AlphaFold in 2018, and it was using CNN, just a standard CNN, same as in computer vision, to predict a 2D histogram. So basically, it was, instead of classifying images, it would predict the heat, or it would show a heat map of predicted distances between every pair of amino acid. So it would, yeah, you could go also in a 3D with every image. You have a chain, and then it gives a prediction of probability of how far these two amino acids are from each other and are they interacting. However, they had also a separate algorithm that folded then the chain to match this distance, and that was using some gradient descent algorithms that could fold the chain that would satisfy the histogram constraints. So the score jumped from 40 to 70, but the threshold of 90 was not there yet. For this, there was a key limitation that one needed to predict distances and then fold separately. And this is something that that has been solved with AlphaFold 2, and that has shot the prediction accuracy from 70 to 90 with AlphaFold 2. And for that, they needed to use the transformers instead. Yeah. Based on basically on the training data that they had from the crystal structures before and the sequence alignments. Yeah, so I will talk a little bit more about that later. So why CNNs are not enough to resolve a crystal structure so well as a transformer, for instance, could? If you look at this image of a puppy on a left panel, as you learned in the week 2, I think, about the CNNs, CNN sees only a small part of the window across the image. You have a receptive field, and each position is then sliding throughout the block. But if you look at the right panel, this is what the attention is then doing. It can simultaneously focus on more points on the same image, and it doesn't need to kind of slowly slide through. So imagine why this matters for proteins. If you have 500 proteins, and even if you're sliding by 5, it takes a lot of time to come and realize that amino acid number 5 is folded and actually interacting with the amino acid 5 and 500, right? So it's important to know that then maybe if the 500 is touching, that maybe amino acid 480 is also touching the space. So one can focus on several regions in the same space. So the visual transformer or transformer can actually, with attention, look at 5 positions at the same time and store this information simultaneously. It's like kind of, yeah, if you would do a game of telephone and go from one to another, like it would take a long time and the information maybe gets lost, whereas with a transformer, every position can be taken into account at the same time in parallel. And before we look at the architecture, we can maybe have a look at a sentence analogy. You have maybe gone through this already in the previous lecture with Christopher about large language models, or it's... Okay. Okay. So I will try to give you some intuition about the transformer with the sentence analogy. If you start with just a simple sentence, the animal didn't cross the street because it was too tired. We know what does the it refer to. The model will use attention to check every single word and see that the tired connects to the animal. So it's simultaneously checking each word, how it's interacting with each other. And when the it is like, it's like the word is sending a signal to every other word in asking, are you relevant to define the meaning? And if yes, then it gives it a score. If it's not relevant to give a meaning, then it gives it a lower score. And this is called self-attention. So basically, every token can, every token looks at every other token to collect the context of the meaning, which is working very well for proteins. That is you can think of it as a very long sentence of amino acids, and they are not just random to each other. They might be relevant to each other in a 3D space. So also, similarly, if we change just one word from the animal didn't cross street because it was too wide, we know immediately that the tired too wide, it connects to the street, not the animal didn't cross because, yeah, okay, the animal, yeah, maybe I didn't choose the best. If the street was very narrow, or if it was a bridge and it was an elephant, that could be a difficult problem to solve on spot, but let's say that the case is.
The answer would be that it refers to animal. If this is trained on the same set, it could
no. Because you don't train the model on only one sentence. This works if you have a large corpus and then it learns in which context it refers to animal, street, and it refers to, don't know, trunk, whatever.
Yeah, and it's then updating with each iteration. Like,
it doesn't keep the eyes for is there an impact? I would say is communicating which answer was bad, giving the
yeah,
increase the weight of the
yes, it's giving.
Next stage that in this case it will give you that.
Yeah, it's actually there is a lot of theory behind this, and like you actually give weights to each of the token, let's say each of the word, and then you recalculate, and then you go further where you look also at the position because position matters, and like if you add a don't in front, like eat pizza or don't eat pizza, like it has to also incorporate all of these other possible semantic meanings. But this is much easier to do with such models that would be with CNN, that is essentially more or less linear in how it works, like linear sequential, so to say.
Attention mechanism. So if you look at the first sentence, so the it is basically going at the end. So it comes forward and the it refers to tire, then back to the animal. But here it goes to the front. So it goes back at the end, why? And then it goes back again at the front, which is the street. So it depends at which position also they are in. So it's like that's the essential idea. We are very sad, because it was supposed to be taught with
Expert in large language models, yeah.
Again, but I think that's the main point, that you go over all the tokens and then you try to find which one refers to which word specifically.
So the times are minus one.
That's right. That's right. This is a very good point. So the calculation goes as power of n squared.
Mm-hmm.
Exactly. Yes.
But there are techniques which allow to reduce this n squared.
Yes.
Well, in this case, n and then minus one half because the number of n is small, so that be exact. But they run this neighborless trick.
Yes. You can go to n log n.
N log n. Yes.
Yes.
With some factoring.
Right.
N log n.
Right.
So these things also, I think, are used at least in the context of the looking for distances between the RNAs.
That's right. Yes. Yes.
So
No, it's great. A great clarification because this I was thinking if I would want to properly introduce it with the matrix and weights multiplications, this would take, I think, a lecture on its own Yes. So I'm doing a very soft, and yeah, I apologize because I'm doing a very high level view without the actual equations and how the weights are put in. But yeah, the context is like this, collected from every token, in this case, let's say amino acid. And in self-attention, we should talk about queries, keys, and values. It's what we kind of already touched. Imagine like each token is generating 3 vectors. If amino acid is a person at a party, uh, it would come there and ask a question. I'm looking to find someone who is hydrophobic and close to me in 3D. Then the key is the name tag on another amino acid that is saying, I'm charged and sitting on the surface. And the value is the actual interesting information that they carry, so the structural context. So all of this, like if you're in a party and you go around querying, you give more value to the, so to say, more value to the to all the queries that come with high relevance. So attention score is basically a dot product of query and key. So the higher the dot product, the bigger the relevance to the later equation. And then there are some functions that are used to normalize, like using softmax to get the weights between 0 and 1. And yeah, the output will finally be the weighted sum of those values. And the good thing is for these all LLMs or the transformers would be that they're computed in parallel, not sequentially, like a residual neural network or the CNNs, where each calculation needs to come to the end, but they can actually span attention across several. Sorry. What is a token? I will come to that in the next slide. Yes, yes. Yeah. So yeah, this is just another query, hydrophobic, yes. I'm in the core, you get packed into the core. Hydrophilic, you'll probably in a reference to something. Well, good. You have some, sorry, what?
Behind. So you are assuming that just these two vectors and one scalar is enough to describe that token.
It is not enough. They have additional mechanisms to, and I will explain them. Like it's adding up. It's building on several different. So you were asking about what a token is. Like in a natural language processing, a token would be a word. So you would embed a vector and it would have a semantic meaning. Whereas in a bio AI, that would be an amino acid, would be a token. And in a case like here, for standard amino acid, we would have, let's say, 7 atoms that would present 1 token. Nucleotide, the same. If we have modified amino acid, we would take each atom as a token. And same with modified nucleotides, and ligands also represent each atom is a token itself. So this is like how it is encoded, and then it scales up depending on the system you're using.
Take the final result very strongly, because it's the way how you discretize the problem, and it means you will operate with different number of precise scaling will be different in each case. So what is doing in terms of biophysical problems? Very interesting. Go on.
Yeah. Maybe like I can say that what makes maybe that somewhat it does not actually answer your question, but hopefully later on I can. So in self-attention, so in neural language processing, every residue checks relationship to every other residue, but AlphaFold also keeps two other representations. So one is per residue, per amino acid residue, for instance, or per token, if you want to say. What is this residue? So it's like, what is my identity? What am I like? The other is the pair representation that will be how are I and J related? And this is it usually described as a proximity in space. So the pair representation is actually the evolving structural hypothesis, because it's not enough to know about the sequence, but you also need to know for the coevolution, and I'll come to that on the next slide. Yeah, I think it works. Yeah, because that will be giving this pair representation that can be then stored in an n by n matrix, where each cell in the model is actually a current guess about how two residues relate in 3D space. So there is like a number of those. Mhm.
Collection of atoms which are connected with covalent.
Yes.
Then it was discretized. Now you can discretize at the level of residues. You still have all residues interacting with all residues. But once you do this, means you reduce everything to the pairwise interaction.
Yeah.
This is a very strong reduction. Nothing bad.
Yeah.
It has to be realized. So I'll put pairwise. Okay.
Yeah. Yeah.
It's good to say this.
Yes. Yes, because otherwise, thank you for seeing it from a biophysics perspective, because I think you would do a great job. Like, um, no, it's great to complement my to say my lecture with a physical and biophysical knowledge. So before we can go to architecture, it's also important to talk about multiple sequence alignment, which is kind of like taking or looking at evolution as a teacher for all of the sequences to come. So the idea is that evolution has been running these experiments for billions of years, and we can have the same protein, be it hemoglobin. Let's say that this is a sequence of hemoglobin in humans, a cat, a whale, and we can see some positions that are very stable. They are important because they're stable, and they are actually carrying that the main structure and form is not changing, and that it doesn't change how a certain function will be done. However, there are some positions that are, or some residues, tokens that are co-evolving together, and this information is very important, because that means if this amino acid will change, you cannot like exchange an interaction that is hydrophobic with a hydrophilic amino acid. So they need to change together to be, for the protein to be able to function forward. And basically that's a built-in signal for the 3D contacts that the models are using, or at least AlphaFold is using it, and other models might be using it through embeddings instead of doing multiple sequence alignment. So the richer the sequence pool is, the more sequences you can compare your input sequence against everything that is known, the better the prediction is going to be. Once it gets lower to 30 sequences, the accuracy drops significantly. So if you're working with a very unusual protein, you have to take it, maybe with a grain of salt. It's the prediction will be poor. And then there is a multi-head attention, so multiple perspective. This is another thing that one can mathematically formulate. We can think of a single attention head as if each amino acid at a party would ask one question, or if you would just talk to one person. However, in protein, many types of relationships are important and all at once. So you have the hydrophobic contacts, you have hydrogen bond, you have covalent bonds, you have co-evolving signals, you have backbone geometry, you have torsion angles, and all of these needs to be simultaneously calculated for the equation to be correct. So it gives each token 8 to 32 questions to ask simultaneously. So this is like then collapsing all of these matrices, reducing representations, and taking that, because each of the head has its own set of queries, own set of questions. One is asking, are you hydrophobic? One is asking, what is the angle between the two, and so on. So the outputs are then all concatenated and linearly projected, and this is actually the AlphaFold score, how to say, core workhorse. It's called the Evoformer, and it uses this multi-head attention extensively throughout the structure prediction. And there is one more thing that is, I think, also important for the physicists. Permutation invariant. So attention, if you shuffle the tokens, the attention scores are the same. Basically, the model doesn't inherently know the order. And that's the same also if you look at a sentence, right? It could be random words scraped together. But for proteins, they are ordered chains. So solution is positional encoding, so that you give a signal to each embedding. And this, I think in Vasvani, the attention is all you need, original publication, that was solved with sinusoidal functions, where each position would get, for each token and each position, you would have a value that is falling on sinus or cosine functions. And this is how you can then control, like, for different levels of attention and position together. So like the importance of the value and of the position. I think it doesn't need to. It doesn't need to. AlphaFold doesn't use it, actually. Yeah. But it can be. Like in the initial paper that is describing attention, it is harmonic, because also it's then a continuous function that you can shift a little bit. But I would not mathematically know how to.
Very nice talk.
Yeah, and also the point is also that the protein structure must be rotation and translation invariant. So basically, if you just rotate or reflect it, if you have a single protein, that doesn't disrupt the process of generating a structure. So that is achieved with the that is called invariant point attention, that respect the symmetry is retained. So basically, AlphaFold uses physics built in the architecture, and that it is achieved through very different models, like that I'm trying to describe piece by piece, but it is a lot of different, how to say, a lot of different modules coming together. I am going faster than I thought, but I think this in the end is only good for you. Now I will go into I was talking about some of the ingredients so that it's easier to understand the whole architecture. And I think after that we can make a short break, and then I will have some case study and what can be done with, for instance, AlphaFold. Oh, it's 2. Okay, because I thought that I started at 1 and I thought it's 1:30. Then it's perfect. Wow. So here is the whole architecture of the AlphaFold 2 as was published in Nature in 2021. Yes, 2021. So inputs are coming from the left, and basically that an input is just an input sequence. And then the MSA and any structural information known from the structural database are going into the model. So the genetic database is performed with the MSA. So we do multiple sequence alignment, and then there is a MSA representation of all of the possible amino acids that could be sitting at a certain location. On the other side, there is also structural database search. So how these paired multiple sequence alignment is connected to the templates that are known in the PDB database, for instance, be it 120,000 structures or so. And
After database this, so this both sequences at same length, or no? They can have different lengths.
they can have different lengths. It's a no, not a fold free. It's not that you can concatenate them, so it's not a is that's not me. Okay. Okay. It's not my brain. Then, yes, these are fed into the evoformer, the famous block that I mentioned before. It's actually a stack of 48 identical blocks, which is the heart of the model. It maintains two parallel data streams, so to say, the MSA representation for the sequence and also the pair representation, which is n residues by n residues in the end. And it's actually the pair. One could think of it as a evolutionary reformer. It's the model's evolving hypothesis about what makes sense, about which residues should be together in the 3D space. So after 48 rounds of refinement, the pair and single representations are then fed into the structure model. And this is actually then when the outputs are produced. So we get actual 3D coordinates for the protein. But even that is then recycled back 3 times because it's actually helpful for the model to learn, and it will not get the correct representation the first time. Therefore, it recycles it and relearns if the pair representations are actually feasible. So it repeats it 3 times in the end. So the 2 towers of the evoformer are, so to say, one is the MSA tower for biology and one is the geometry. So, and both of them need to be updated, both for the MSA and also for triangular updates. And I think this is also something that, yeah, I have to tell you about it. Another thing that maybe the physicists will appreciate is the triangular attention and the updates that are also updated in the pair tower in the geometric part. What does it mean? If we have the pair representation encodes relationship between all residue pairs, but not all relationships are physically possible, as you can imagine. Like if a residue A and B are 5 angstroms apart, and then B and C are 3 or 4 angstroms apart, A and C cannot be 50 angstroms apart because it's just like the triangular inequality that must be satisfied. So the AlphaFold 2 is enforcing this through triangular updates. So for every pair, the model always considers all possible third residues and checks for consistency. So basically what the starting and the ending node is. And it's not a hard restraint, but it strongly encourages that the geometric consistency is maintained throughout the model. Because if there was no constraint, no geometric constraint, you could get a lot of structures that are really not plausible because could emerge in the between different residues. Yes. Like if there are, yes. Oops, if I myself also understand it correctly. Because yeah, for each of them, when you do calculations for each of them, then at some point you will reduce the mathematically impossible ones that are too far away, like based on the structural templates and the MSA. So all has to be updated. It's a three particle distance. What is?
Set a number that is descriptive for the distance somehow of distance between the particles in space. There were such things. I am not.
Was that very recent? Like, okay. I'm a bit surprised if this is like what can take more particles. I don't know. So now the final model is the structure model. We need the abstract representation as actual atoms in 3D. So Evoformer gave a refined pair representation and a hypothesis about what's close to whom, but the structure model then in the end turns it into a 3D coordinates. So it's kind of if you would start with all 500 residues of of a protein all piled at the origin, position 000, and each residue is kind of like a tiny rigid body with its own frame and coordinates. It has a frame of a position and a rotation. So it starts randomly, and then over 8 layers with this invariant point attention that we mentioned before, each residue looks at the other and it decides I should move 5 angstroms away from you or in above each other. It's physically impossible. So the whole, the physics here is elegant and probably beyond my understanding, but the whole computation is then invariant to global rotation and translation. Yeah. The tour.
So then you also know the balance angle is fixed, and then the distances are also more or less fixed.
Fixed, yeah.
Three is the collection of torsional angles. For the polypeptide, a couple of torsion angles by side per residue. And then you just reconstruct the whole thing. It's just reconstruction of the chain using these two things. That we know the balance angle, which is the angle between carbon and carbon.
And the carbon hydrogen, yes.
The distances are also fixed.
They're almost fixed because they're learned from the attention and the structure templates. Yeah. Yeah.
The balance angle temperatures fluctuates about 4 or 5% of its value, and the distances fluctuate with 2 to 3% of their value. Distance. And if you know all these things, you can reconstruct them. If you have the sequence of amino acids, you can put the sequence which are side chains. You can put side chains if you know the sequence,
Yeah.
then you will get it flat.
Or you can, or you rotate it, yes.
And then you can take it and everything. But there are numerical problems when you start making, when you go to the local system, this transformation, this rotation and the translation, these kind of transformations are very much prone to the random error accumulation.
Yeah. The what? But the rotations, you said.
Yes.
Okay.
When you fix the distance, fix the balance angles, after some time, just because the accumulation of numerical errors, very
Yeah.
close.
Yeah, because the error accumulates from different modules, but
Many like summation, so matrix multiplication and summations. And when you do this, these are rotations in space.
Yeah.
So many times when you repeat this many times, then what happens is these numbers of which distances, let's say, are represented, although it's what it is, 5 times 10 minus 15 or whatever it's done.
Yeah.
Then these errors of multiplication, they get accumulated.
Yeah.
And particularly this algorithm is when you recalculate the position from local coordinate system, is very much prone to accumulation of such errors. And it has to be very carefully checked every time when it's applied. Because if the errors are random, in principle, they should cancel each other. But
Yeah.
in certain, it's just numerical thing. In certain algorithms, they are prone. It's fast Fourier transform.
Okay.
This kind of this local transformation are prone. I'm sure maybe people know already.
This I would not know. It's beyond what I can say, but yeah, it's definitely
very much taken whenever applying each piece of this very complicated framework. And that's really has to be very careful, starting from the processing the PDB database entries, because the structures that are recording PDB for the same molecule, there can be hundreds of structures, and some of them are really different from each other. So it has to be, one has to be really careful with every piece of it. So all over how it works, I'm amazed that it works.
That is true, and we had we had a similar problem in our benchmarking, like already when you're selecting data from the databases, you have so many crystals, and then, that's maybe an asymmetric unit into the PDB, but not a full biological assembly, so the properly folded protein, and you download that and you use that as your ground truth, then basically all your assumptions are wrong. It is a lot, yeah. And then you have missing residues in the structure. Also.
Parts that are not visible.
Yeah.
They have functional.
Yeah, and this is still an kind of like unsolved. Okay. Yes. Sorry, online. We had yeah. Okay. Thank you for commenting on that. Let's go on to maybe just one example that, like, at the beginning of the structure model, all the residues are placed at the origin of coordinates, as I said, and then every step of the iterative places, the AlphaFold 2 is reducing a set of affine matrices that displays and rotates the residues in space. The representation does not reflect physical or geometrical assumptions and can be and it can undergo a lot of geometrical assumptions. No, it can generate, sorry, not geometrical, but structure violations. So they are like a lot of them are wrong, and this is particularly visible, like in the folding of this protein, like the trial and error of the model to put the certain units or subunits of a protein together when it starts from a mass and then it tries to satisfy constraints of coming from different parts of the model. Yeah, you can.
It's like go step by step, piece by piece. That's not so good. It might be that the minimum is not close. I mean, the fun over the energy space, the energy, say surface, that you are locked in one local minimum. Well, local minimum could be far apart, and then I'm sure there are ways.
Yeah, I was whether the search is local and if the global or local minimum of this energy funnel is satisfied. I am pretty sure. I am actually not sure of how to answer this to you, but one can test that then later simultaneously, maybe with molecular dynamics, where you do simulations of how the molecule is behaving thermodynamically over time, or at least in binding.
That's sort of global minimum.
Yeah.
It's not accessible. It's also a problem there.
Yeah. So yeah, in search of the global minimum still. And I said already before that AlphaFold doesn't stop after one cycle. It does 3 recycles because some errors only become apparent once you see the initial 3D structure hypothesis. So the first pass, the model might not realize it's done a mistake, and then it can update and fix the geometry in the next iteration round. And basically with the CASP 14 result, like at this competition, I think the result median global distance was around 92%, which is getting close to what also the structural methods can achieve. It was actually with that CASP declared the folding problem is solved for single chains of proteins. So that is like, because the single chains are generally should not be disordered. Disordered proteins are still very problematic. Antibody antigen interactions are still very difficult because you have intrinsic variability of the immune system. A lot of permutations that are not represented maybe in the known proteome. And that's where, because it's kind of a site of active evolution, it's hard to get the model right. And lastly, what changed in AlphaFold 3? AlphaFold 3 came out in 2024, and it came as a major paradigm shift. Why? Because the scope, first of all, expands from a single proteins to entire molecular complexes. So it can do protein-protein interaction, not just one protein. It can do protein DNA, protein RNA, protein-drug interactions, all in like one run. And architecturally, the evoformer that we talked about before is replaced with performer, which is just simplifying the MSA processing step, which is computationally quite slow. I think it's using hidden Markov chain models to search through this space, and that can be computationally quite demanding. And here it forces more information through the pair representation block instead of through the MSA model itself. And also the biggest change in the structure model is that it's using diffusion module. There is no rigid frames anymore, but it starts as a random atom cloud that it's trying then to denoise using all of the information, similarly as AlphaFold 2, but not starting with the rigid, but kind of like as a cloud of theory. Are you? Yes. So, okay, Suva will teach more about the diffusion theory in the future, like how this process is. Is the basic for, let's say, the newer models such as AlphaFold 3 for structure determination, but also for de novo design, where you start the inverse problem. You have a structure and you say, give me something that binds here and has this certain 3D structure. So there, this denoising and diffusion modeling is it's much more flexible to acquire a certain, let's say, chemistry. However, even there, from experience of the colleagues, it seems that this is very biased to the trained data, that what one can come out is not something that was not globally yet seen from evolution. And this is something that I'm also wondering a lot about, how much can you push the system to be out of natural, like what evolution has processed? Can you really come out with novel sequences if your baseline, or not sequences, sorry, structures and sequences maybe, if you trained, your ground truth has always been in the world of what is physically possible by evolution. But this is more maybe a philosophical question. I'm not gonna, um, we can discuss this in the break. Uh, yeah? Yes. It it will be also a 3D structure. Yes, so you will get, for instance, I will show some examples later. You will get, for instance, if it's a protein, yeah, you get a protein backbone and you will have a ligand or just uh, an organic molecule bound to it. Yeah. Uh, yes. I want to point you to this illustrated AlphaFold blog if somebody is interested in to diving much deeper. I have been reading it, and so the authors work from Stanford University. They've done like a marvelous job illustrating the steps of AlphaFold, where you actually have an interacting table of content. Very much detail. It would take hours to go through just this architecture. So like I can really recommend that. And the other one is the illustrated transformer. It's also a blog that I was using a little bit for some explanation. So if you want to learn how the embeddings and tokenization work, the illustrated transformer is also a good web page to go to. Yeah.
Two questions. The first one is that the diffusion module is essentially a bulk of things, right? And then you have a back propagation to predict that something new, or you have something supervised to predict whatever you want to predict. So in this case, so the prediction is unsupervised or supervised? I guess it's supervised.
Uh, I am not entirely sure because I do know the different, like even if you start with a different seed initialization in the model, you can come up. We have tested this in our paper with completely different trajectories. And for instance, when we compared our structures to the original crystal, or what is the ground truth structure, we could see that if we choose 5 different random seeds, depending on the starting path that the diffusion model starts, and probably then the energetic route it is taking, where it's being guided, you can get a wrong model entirely, like not physically acceptable, to one that is docked with a 90% accuracy. It's mind-blowing. It's also, for the study that we did, AlphaFold 3 original paper for antibodies and antigens, so antibody protein, they suggest that you actually run 1,000 different seeds. I mean, the most computationally difficult step is the MSA calculation. Once you calculate a new trajectory, however, we have also experimented with running, you can run one sample 10, 20, 50 with the same seed or a different seed. We saw that within the same seed, the structure might improve like 60%, but the model, internal scoring, doesn't know about it. So there is a big disconnect because it's kind of a fixed, we call it in our paper a fixed mindset of the diffusion model. It's starting from the, probably it's coming from the evolutionary performer, that this confidence information is locked in. The model might improve itself, but it doesn't know it's improved itself. And this is, yeah. Yeah.
So what actually is going in from the layer which was the one and the 3 arrows that goes again?
So from the performer, what goes into the diffusion model? Yes, from performer. So
yeah, seems.
Yeah.
Yeah. So these are some extra information to the network.
So I wonder, like, this is not clear to me, because I think this would also answer the question why the information is not updated. I think it comes in only once at the but the architecture is still not released. And then it works with that, but it doesn't get any ground truth from the outside information to know it has improved. Like, this is how I understand it. Yes, this is internal scoring and clashes. It has many like internal functions that do that. It also gives you output as like if it has clashes between atoms and how many structural clashes they are. And those that taken out or ranked very poorly, or like they're lost in the different module blocks. But
Really complicated software and Singapore's. I have, it's a great, I mean, it's a great, great method of
great tool, yes.
very carefully applied, especially so complicated problems. There is a lot of jargon introduced. Well, there is a very well-known terminology in the field.
Yes.
It's not used. Now I am afraid it's not used because they are unaware of that, or they don't want to speak about that, or there is something going on.
Yes, I don't know. I think the problem is twofold. One is that it has had an exponential growth in the number of models and people working with it, and it's not a necessity that you understand it to the It's like, I'm not saying I understand it to the core. It's very complicated. But I think it's a mix of both because there are so many tools and the evolution of the tools is so fast that the field is not kind of updating itself. But this is dangerous.
For instance, I learned that one tool I learned was Gromax.
Yeah.
Gromax took me a year and a half, 2 years to learn. I still cannot say I know everything.
Yeah.
How each of the model is working, functioning, what are the drawbacks behind what is the model. DFT, I'm still learning it, but
Yes.
more or less I know how each of the pieces more or less how they work. Now here it's one level of complexity more. And when it's like this each details you see, when I go deeper, I see things that I don't.
That it's a very I think it's the speed of how everything is running that maybe people have the idea that it's I don't know.
Then it's not a development. Then we should abandon science and rely on our chemistry.
Yeah.
That's the for me.
Okay, I have two more slides and then we can have a break. So basically, side by side, just quickly recap difference of AlphaFold 2 and 3. Input for AlphaFold 3 would be just sequences for proteins, but now it also takes proteins, protein DNA, RNA, ligands, ions. Trunk was evoformer, now it's optimized performer. MSA usage was heavy, and now it's simplified in the newer version. Here it's rigid frames, IPA torsion, then diffusion as a core atom level denoising for AlphaFold 3. And yeah, the key metrics is that for the protein ligand docking, there was a 50% improvement with diffusion conditioning because the IPA intorsions work very well on the backbones, whereas ligands, they don't really have backbones. So that was a huge improvement for protein ligand interactions. And as you may know, AlphaFold 3 is only open license for academic use, and it has quite a high standard accuracy. However, a lot of software that is quite AlphaFold 3-like has emerged. MIT open license of Boltz-2 that also predicts affinity, but there's also a lot of problem there because how do you define affinity? How is it measured? And it's poorly correlating with experimental data, especially in the field of antibodies. Then we have ASM-fold, mostly for proteins. And yeah, one of the open license ones. And Rosetta-fold, of course, from Baker Lab as an alternative. And with this, I think we have used the first three in our benchmarking that I'm gonna introduce after break, just like shortly what the results were, what we tried to achieve. And now I think we're quite on time actually for the coffee break. So should we come back 250 or you need more? 255. Okay, we come back at 255. Yeah. Okay. Okay. Yes. Yeah, otherwise we find you. Thank you. Yeah.
Sorry for
Should we be slowly getting back? Do we finish on time for the Easter? Can people online hear me?
Yes, thank you. We can.
Okay, thank you. Good. So let's continue that we have time to, color your Easter eggs or whatever it is that is your joy for this weekend. Okay, so now I will talk a little bit about the application part, about the benchmarking case study that was part of the work that I have done in a collaboration with 2 other groups. So basically, can AI tools distinguish real binders from fake ones, like from decoy ones? If you just create a scenario where you have a nanobody antigen. Yes?
We take into account interactions, right?
Mm-hmm.
Does it also take into account plus protein, like I don't know, conditions dependent on the environment factors like temperature, pH, so let's take that into account.
Good question. So for the online people, the question was whether AlphaFold takes into account also the interactions such as pH and temperature and so on. I think you can, pH for sure not. Temperature, I think you cannot take into account, but I know that there are some cases where you can work with at least with different conformations of protein where they are in this hollow or what is the other mode? The hollow and sorry. No, the hollow, like the hollow mode, like the hollow or like open, like different conformations. So no, I will look it up and tell you later. Yeah, but it's still like different conditions are not taken into the account, like the pH and everything, because this is also a big question, and like how to stabilize, for instance, intrinsically disordered proteins or proteins that are stabilized upon binding, because then the conformation changes very often. And that's also a problem with enzymes or ion channels that change the conformation upon ligand binding. So this is still, it is partially solved. It's working in this direction, but I don't know of cases that you could use that for now. I know that like in molecular dynamics, you can work with that, with temperature changes. I don't know, you cannot affect you cannot work with pH changes in molecular dynamics, not really. You cannot, but temperature for sure, pH, ions, yeah, addition of ions, yeah. So, okay. The idea was like, if you have a tool that can predict complexes, be it protein and protein-protein interaction, specifically nanobody antigen, it should be very specific interaction. So if you feed in real case study from, let's say, real binders that have experimental structure resolved, and you then take from these binders, like nanobody antigen, you take the antigen and you take all the other binders to it, so you make shuffled and not real pairs. Will the tool be able, with its confidence score, say this is probably not a real scenario? Like, how good are they distinguishing real from shuffled decoy ones? Just a few words about nanobodies. Nanobodies are these small recombinant proteins derived from camelid heavy chain antibodies. Human antibodies have this for IgG at least, you have a heavy chain and a light chain, and the binding to the protein or antigen would occur here on this region. Whereas camelids like llamas, Bactrian camels, alpacas, they have next to the heavy chain ones, like next to the normal immunoglobulins, they also have those that are derived of light chains. So during evolution, for some reason, they have lost it. It's not clear yet why this is the case. And the nanobody is actually just this tiny part, like just the circled part, also called VHH, that is like very small, and it's only the binding region of the whole heavy chain antibody. It is roughly 10 times smaller than a normal antibody, and the binding occurs with these loops that are called CDR loops that are very prone to, I mean, they are differentiating a lot upon affinity maturation. So upon meeting the antigen, like when being exposed to a virus or bacteria. So these are evolutionary, very not unstable, but they evolve a lot. The advantages are that they're small, they can reach cryptic epitopes, so cryptic hidden binding spots. Epitope is a binding place of an antigen on a protein. It has better tissue penetration. Traditionally, how one would traditionally obtain nanobodies is mostly through animal immunization. It's the same way one would get normal antibodies. So you would introduce a protein or like a viral particle into an animal and wait for 2 to 3 months that the animal starts producing the immune response to it. The extraction library, clone it into the phages, and then actually expose the phages to those antigens in a plate and see which of these nanobodies are binding, and like this come for several rounds of selection to come to possible nanobodies that one then actually produces and with different physical and logical methods. I'm sorry, I'm going to use it like this, I hope. It's not guaranteed that it's computationally screen and do kind of, for instance, bolts one, to find possible pairs of nanobodies binders against antigens, so against different proteins that they recognize. And for that, in our lab, we're using a naive library. This means that we did not immunize the animal. We just collected blood. I mean, still, it still is an invasive method, but we try to work with how to say basal a starting system for immune system, starting ground for immune system for selection. And in the study, together with colleagues at Oslo University and Cambridge University, we did this Champlu method, that's how we called it, where we did nanobody antigen interactions like modeling. And the truth was we took 106 experimentally validated complexes from the PDB, so from the protein database, and we would take out the sequence from a protein and from the nanobody, and then we would shuffle them. We would bind each one with each. So we did like this matrix where in this round we always have a nanobody, and here we have a pair antigen. And only those on diagonal are real ones. Everything else is a decoy, and it should not be a binder. So in total, we had 11,000 roughly negative controls. And this, I need to also say, is very uncommon. People usually always validate their methods on real binders or on real complexes. And they say, our method is 5% better, or 2 or 3 A or C. You know, you can find so many precision matrices to say how you are better. But I think there is a lot of failure, or like a sort of like ignorance, to also provide enough of the negative examples to see what the actual ground truth is. We tested 3 tools, AlphaFold 3, Boltz 2, and Shy. And the question was, can the models assign higher confidences to real binders than decoys? A solved problem, this should not be too difficult. Uh, Hello? Oh, now it's working. Can you hear me now? Yes, okay. Sorry. I totally didn't notice that I'm not talking. Do you wish me to repeat this slide or the previous slide? If you can, yes. Okay. I hope you don't mind. I'll just quickly go through the experiment design. So I was saying that the challenge in the benchmarking is if we want to know, if we take a nanobody and antigen, nanobody being the binder and antigen being the protein, these interactions are key for drug discovery, for immunotherapies, and for vaccinations, for instance. And there is a big promise that you can just screen the nanobody or antibody libraries and get super good drugs very fast. What we wanted to test Do models assign high confidence to real binders than decoys? For instance, if we take 106 experimentally validated complexes from the protein database, and like they are presented here, the nanobody, if you take just the nanobody out, and on that side you have the antigen, and the real ones are only those that are on the diagonal, okay? So this should have a high confidence from the model, and everything else should have a low confidence. So this was like our benchmarking test to see whether we can also reliably use it in the future. Because the problem in the field is weekly almost, there are new methods coming out. People claim to have better confidence scores, better accuracy. But the problem is also what you test it against. If you always test it against a real binders and you don't include possible negative controls, then I think you cannot really say for sure that your method works if you want to do a prospective screen. And what I said before is this is an example of how we were evaluating our results. We would, at least for the real binders, in yellow here you can see a crystal structure from the PDB. And on the right upper side, there is a nanobody bound to this calcium channel. And the blue is actually bolts2, so machine learning model predicted structure. And you can see that in this case, when you align the two the two 3D structures, the real and the predicted ones, you see that they're basically one-to-one. The confidence is above 0. And for the dog Q, which is the scoring between the I. So this not always the case. In our experiment, we saw that for the free tools, AlphaFold 3, Boltz2, and Chai1, we cannot really computationally distinguish between real and shuffled complexes. We sort of see a diagonal forming for AlphaFold 3, but there is also quite some other highly confident decoys. If you look at Boltz2, and there's been a lot of promise for Boltz2, and also, I mean, it is a great method, nothing to say against that. However, there is not even a slight, even if you want to imagine, you don't see a diagonal there. So it means that it's equally confident about decoys that it is about positive control. It's not a binding pair. So when you look at the structure, but you know that it is not. It's kind of like you have you don't. And positive risk. So if in each case, it's 91 for bolts. I don't. Yeah, maybe we can just turn it off and I will. Okay. Can you guys hear me online? Okay, thank you. And sorry about this. Yeah, so you can see that the number of false positives is really high also, but even if we go to very high confidence threshold 0.8, then we recover just a few, like 1/3 of true positives, and we get a lot of, or at least, okay, in the hundreds of false positives, whereas for bolds 2, for instance, we still have over 6,000 false positives and so on. So this was quite of a surprise for us. Also, we found that there is a disconnect in the confidence. The IPTM, what I was talking about before, is the interface of the predicted interface of the model. So model internal confidence does not correlate with DocQ, which is the actual quality score, meaning that if we can have, like for AlphaFold 3, quite a high DocQ score for AlphaFold 3, but its model confidence is very low. So basically, it doesn't know that the model is actually, it doesn't know itself that the model is doing well. So here we have a lot of false negatives, whereas for bolts 1, this is like a DocQ score on a y-axis. 1 is like experimental structure, and 0 is like very poor. You can see that you get quite some false positives that you filter out. And this is actually looking just at the real 106 ones. You cannot do this alignment with the decoy, because for decoy, of course, there is no crystal structure because it just does not exist. So this is problematic because if you, for AlphaFold, if you put a threshold somewhere here, you will miss quite a lot of good binders maybe, whereas for bolts, there is no threshold high enough to not get also quite a lot of false positives. And Chai is also quite underconfident in this. So the confidence score of a model is not really reliable to score.
Did you publish this?
It's on the bioarchive, yes. And let's say that we're somewhere in revision. We don't know how it's going. It was quite a lot of interest from the community because we released, there's a lot of background that I'm not telling here. For each of the model, we ran 50 samples. So this becomes 3 models times almost 15,000 times 50. We're at, I don't know 2 million structures roughly that we produced. And there are a lot of false negatives. So it took a lot of compute, and we are making it freely available for the community to learn from, what is also not real. So we'll see how it goes. It's bioRxiv. It's in some fields it's considered published, but I think it's going to be a long way to come through review. The third sampling, what we also did was for the real structures for which we had the ground truth, because for decoy we don't, right? We have looked at what happens if we sample deeper. So instead of just running 1, 10, or 50 as we did in the whole matrix, does the structure improve? That was one question. The second was, what was the energy? How much energy did go into these predictions? What was the gain in information that we got? And was there one other? Yeah, what we saw is that the more samples help, the model doesn't learn that it's getting better. That was the second thing. But also we are very sharply getting diminishing returns. So like the biggest jump in DocQ, so in the gain of a structural quality, is in the first 10 runs, or 10 to 25. And then we're actually just having marginal quality gains in the subsequent transitions, apart from the Chai-1 model that behaves a little bit differently. Also because it's working with embeddings, and the architecture is quite different in that sense. I mean, at least the steps are a bit different.
You know, it means actually to me that the scoring function is not constructed correctly. So the scoring is based on the score which you decide whether the thing go in the correct direction or not.
Yeah, but that's then within the model itself. Yeah. And I've seen that there's been papers that are now trying to kind of force or do the ranking within AlphaFold to give it a better score, informed from, the real world scenarios. But the model itself, at least at this stage, they don't really have the ranking you can trust. Like, at least not. The level of trust is kind of correlated with the amount of money and hands that you have to then do the experimental validation, or how many robots you have to do the job, because at some point it becomes impossible to check all the false positives. So yeah, the recommendation is like a stage workflow, not too much sampling, but also deep sampling for high value targets only. Like AlphaFold 3 recommends you actually, for nano antibody antigen, you run 1,000 seeds. And this is really, it's inflating a lot the number of samples and scorings, and even there will it really improve? Because the model doesn't know it improves. So what does it help to run 1,000 runs if you get a high confidence and it's wrong? So these are all like the questions in the design and the scoring functions that the community is working hard to fix it. And then just a little bit, maybe shortly, on a practical side, I don't know, are you or any of you using these tools, AlphaFold or CollabFold, the open servers for your work? No. Okay. I will just show some examples, like how you could maybe use it. But then if you have other questions, like, you can come back to me via email. I didn't put it here, but it's my name, surname @UNG, or you can Google me if you have questions after this lecture. So AlphaFold server is a good place to start, because it also has a visual interface. It is, you can, if you have a Google account, which is necessary, you can run up to 30 jobs per day on the server, which is already quite good, I think. Boltz 2 is And I have to say for AlphaFold, for instance, I got a license and I can then also run it on the Vega cluster or on HPC. So there you can then vary a lot of other parameters, much more than just the standard constellation that's in the on the server. Boltz 2, Chai 1, you collab fold, you can get all of them through the GitHub. And maybe let me tell you something about the confidence scores if you were to run a molecule on the AlphaFold server. One of the key things to look at when you get the crystal structure is the PLDDT. I always have problems saying that. It's predicted local distance difference test. It tells you per residue confidence, because the confidence scores I was telling you about before, it's different scoring functions working together. And this one is, if you want to look at the complex at a very specific residue, you will see how big the score is. So like if it has over 90 accuracy, dark blue, the model is very confident it's correct, and it usually also is on this in this case. If it's less than 50, it's probably disordered or hallucinated. And disordered protein, you can see here in orange, is this like loop that the model doesn't really know how to fold it. Then it's also IPTM. It's a predicted aligned error. It tells you about the interface or the complex confidence. And also above 0.8, it's a very high quality. And one can look at this also on the AlphaFold server. Let's say you just put in a sequence of a ferrous ion transport protein, one example, and you get out a 3D structure, and then within the AlphaFold server, you can already get this matrix, which residues are low confident, which are high confident, and this map is telling you which interactions at the interface are high confidence. So the difference in the alignment is very small, so they're correct. And in the lighter regions, it means that probably that structure is not very reliable because it's just deviating too much from the real, how the real interface should be. And then this is an example of a very difficult problem. What is it? A mediator of DNA damage. Checkpoint protein 1, which you can see here on the bottom. And you can see that only the core could properly fold and everything else is a lot of spaghetti. Also here, darker is better. So only this very little part at the beginning is correct and everything else is massively wrong, which, I mean, as a biologist, you see this and you know something's wrong. Maybe also to you, it says something that this is a very uncertain prediction from the model. And this here, I have to say, I forgot to acknowledge the resource. This is from the AlphaFold 3 paper. It is one of the very difficult proteins in complex with antibodies that was solved using AlphaFold 3 very correctly. Throughout the model, it's above 90. The PLDT and IPTM is also very high. And you can see in light gray, it's the actual crystal structure because this has been resolved. And in the blue is the coronavirus spike, and in the green are the antibodies, and the yellow are the glycans, so the sugars. And you can see that it's basically, the overlap is really striking. So these models are really, it's like, one doesn't want to criticize them to say, they're doing a poor job, because I think it will need more incorporation of the physics scoring to become closer for some difficult problems. But on average, they are performing very well, when you can see that it's like to experiment at 0.8 angstrom difference. This is almost the resolution that they can get with the crystallography. So it's getting there, really. And then we can also have cases like this. This is one from our study, where we were shuffling nanobody with mismatched antigen. It should not bind. The green is the original protein, and the blue is the original nanobody that binds the green protein. And then you can see if we put a different antigen, which is in purple, to the same nanobody, the nanobody is placed here to the same, we align it, the nanobody is the same, but we see that it creates a confident structure of the purple one. This is aligned on top. The green one should not be here, but just for your reference. And it is confident 0.9 that this is correct, even though it's a completely wrong protein and the nanobody should not recognize it. It's biologically should not be feasible. So it's a decoy, and it's important to include negative controls to also stay a little bit humble and see what the limitations of the methods are, that you cannot always trust the high confidence score from a model. Then one interesting database to look at and maybe model is the intrinsically disordered regions database, where these proteins are not folding properly, or they are in a semi-folded state, even in the native proteins. So it's hard to create crystals from them, and therefore it's also very hard for a model to predict such structures. And I was going in and taking one of the proteins that is highly disordered. This is, for instance, eukaryotic translation initiation factor for E-binding protein 1, a long name. And you can see that the model is doing quite well in the core, but again, it is there are quite big regions that are disordered, and the model is struggling with. So there are quite big distances to what should be a real interface and what is predicted. And one can then also go within this tool and check that predicted protein against the crystal ones. One can align it, one can play a lot in the protein world, but there are also tools for material sciences, for general chemistry, ligands, ions, metal ions that one can be testing. So the takeaway is that workflows should be staged and multi-tool, preferably. Never really try only one tool and rely on it, but this is anyway a good scientific practice. You will never, or I will not say never, because sciences work differently, but generally you do not in biology, you do not trust experimental results from one resource or one source. You always need to validate across different experiments to be sure about the biology or the signal that you're getting. And we need to know that AI is generating hypotheses. Some of them are good, some of them are worse, and cross-tool agreement in preference with the physical-based method is a good way to go if you want to have accelerated discovery that is also correct and not inflated. So with this, I will actually be finishing. Okay, a little bit ahead of time. I think you may not be too angry. I have used different resources, and here are some tools and learning resources that you could have a look at if you're interested. I would really recommend this Illustrated AlphaFold if someone is interested, and Illustrated Transformer because it's nicely animated. And I don't know if one of you knows the StatQuest guy. It's a YouTube channel of, I don't know, mathematician, statistician that is doing, I don't know, I think you might like it if you like my humor. He's just doing animated through drawings of how the networks work, and he sketches everything on hand, and you can follow step by step, be it transformers, be it, I don't know, backpropagation, you name it. You can spend days in his channel, but you learn a lot because he's really good. It's so I could I didn't post it here, but it's helped me a lot also to understand certain concepts in the past. And yeah, EMBL has an AlphaFold training app where you can actually go through the concepts and test your knowledge. And if you have any questions that pop up later, or if you would want to like set up experiments yourself and you don't know how or what to take care of, you can always reach out. I'm based in Rožna Dolina, but reach by email or we can talk if you have questions. And that's it from my side. If someone has questions either here or online, otherwise I will then stop the recording. Yes? Yes.
It's going to be very stupid because I'm not from this field, I'm an astronomer. So.
I don't know much about astronomy, even
yeah,
after
my
this
question is actually about biology, but not about the details of your talk, which was very nice, by the way, because I was able to follow the absolute majority of it. So you did a good job.
Thank you.
My question is like this. Okay, so we kind of solved with an asterisk mark of solved protein folding. When it came out, it was big news. It was basically we will advance development of blah 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 blah. You know how it is. Now, as I look back at the news that I've read in the last, let's say, basically year since AlphaFold came out, and also with a quick search that I ran in the background, I'm sorry, it seems like the fact that we solved this particular problem did not really speed up the final output of the drug production, new chemical reaction, stuff like this. I understand why it is, because it is a complex pipeline with trials, errors, whatever. I mean, we solved one small part. It was producing tons of papers in the previous century. My question is, what would you consider to be the biggest bottleneck in this pipeline from making protein hypothesis and to producing final drugs or other useful chemicals, or understanding the nature of diseases? And whether this bottleneck is something that can potentially be automatized, like maybe we need to understand better how the molecules behave in dynamics or something, or is it just that there are so many very different aspects that it requires through general intelligence?
Yes, I think it's very good questions. I don't know if you could hear also online. There were 3 questions, right? Yeah. If I roughly, I try to answer all and like also repeat the question. So one was like, what is the biggest bottleneck in really speeding up the drug discovery? There was a drug, I think last year, that came through a clinical trial actually and was totally like from de novo predicted drug against the disease. I forgot the name. But it actually went through all of the stages from de novo design to the lab testing to the clinic and has been FDA approved, if I'm not wrong. What I believe is stopping the advancement is that still, as with many intelligences, I think it generalizes very well. And when you want to go into specifics, this is when the system breaks, because it generalizes so good to predict a possible, binding site, generalizing on all the millions of molecules if you want to see that it has seen. What the majority of models still don't take into account is really the one-by-one chemical interactions that in the end might matter, or that you have in the real world when you were actually, like, you can have a very good in silico predicted binder, but when you will go to the in real world, where it will usually fail, you might have a different temperature, as you said, different pH. You might have access to water molecules that modify or shield certain parts of the molecule. Or when you go then in vivo, it's a third level of complication. You might have glycosylation, so the sugars on the top, they are partially, and they are being solved actively. So it is a sort of being solved problem. But it is a lot of other components that might be reacting in the end, and it is hard to model everything of that into the account. And also, for instance, for antigens and antibodies, or any binder, like when the two molecules are interacting, molecular dynamics will still tell you a lot, because it will tell you, like, if you're increasing experimentally the temperature in the simulation, at what point will the molecule fly off, like how strongly it is binding. And they are modules that one can apply, but if one would want to simulate all of this in a model that is already like pushed, I'm not saying it's pushed. I think it's quite impressive what they do, and it's like, I really need to say, like, the way, I think it's a very smart design how they did it. So, like, really heads down to the whole team that is collectively working on that. But the most difficult is to gather all of these dynamical simulations in, and then you have binding affinities, and then the in vivo comes with its own set of problems. So we have seen a lot of papers in the past where they have libraries, they start with hundreds of thousands of millions of molecules, and in the end, they screen and they come with 10 binders. And then not necessarily, they will work in the lab maybe, but that's then the first step. Most of the drugs still in the clinic fail in the clinical trial, not actually in the lab. So this is the translational aspect that is not, that is being modeled by a different community. But I think this is why it's good to give this, hypes to people, because it gives people hope and incentive to fund these fundamentally important problems that were long time, some of them also stuck, and people frustrated because they are difficult tasks, right? So it's a big plus for the community. But I think generally, that's going philosophical, sorry, we are overthinking of what progress something will bring. It brings initially a big push, like it's like electric cars with AI, with generative models, with CAR T cells, for instance, with CRISPR-Cas. There were promises. Now we're really solving like the cancer or AIDS or this and that, and we are solving it faster. But then the plateau starts again, and then we are chasing the next thing. So I think collectively, community and science needs to be patient in discovery, or incorporate a lot of knowledge, for which AI will definitely help, because it's hard to hold so many information in one brain. But it's not probably answering your question, sort of. But I would say these are the bottlenecks that, like, at least for like the dynamics part, that are like very computationally intense, take hours for big proteins. it's not scalable to millions, like when you have a big library. And what was the other question?
I mean,
It's
it's
like,
all one business.
Yeah.
Whether that's this bottleneck is something like whether it's single bottleneck that can be solved maybe with some other complex design, or is it basically if we had a model that could simulate the discovered molecules or something
Yeah.
like this, for which, but if it could simulate it in dynamics, would it be crucial to speed up this process, or it wouldn't be the major bottleneck as well?
I think it would speed it up a lot, especially for the binders, where you are calculating the probability of stay in the interaction, because you can loosely interact, right? You can form also transient potential bonds, or you can have very strong like hydrophobic interactions and hydrogen bonds, or like you can just shortly come a little bit together with the charge, and it's not a very specific. So if one could, and I think there are people working towards that, I think this will be solved, and this will probably be the next big push. What I am wondering though is, it's almost again a bit philosophical when people talk about de novo design. Where does the de novo start, and where do you start reinventing the wheel? Because I think there's been proteins that they have, there's been a paper I need to cross-reference check with my professor, the boss, it was supposed to be an artificial binding protein system that is like revolutionized the way things are discovered. And years after, maybe 10 or 15, it was discovered that one organism, a fish or something, actually has this protein already in the system. I don't want to be cynical, but I wonder how much of a novelty can you construct that will survive the physical and biological? Because is there a finite universe? Sorry, yeah, this is like.
We don't care if it's true, and we would care if it's true novelty. But if we discovered something useful and then we discovered that it already exists in nature, good.
Yeah, no, I think yes. I don't think like I don't have a problem with that, because I think it's very good if you recycle what has been worked on with evolution for a long time, and it's probably an effective solution that you can try to optimize. And I think that people just like novelty, and it sells quite good to say, yeah, it doesn't exist in nature. It's like, it's out of, the, but I think people are happy to discover also like the natural, anything that works and fixes the problem. Yeah. Okay. I think with this I will, yeah, yes. Yeah. Yeah, so that is actually a very good question. In de novo design, when you design sequences with diffusion, right, you can create sequences that are, at least like for the use in humans, very immunogenic, meaning that they were not selected before in the evolution, or they were deselected because for a certain purpose they would, let's say, induce an immune reaction, or like they could potentially be dangerous. So also the tests, there are tests and developability, and like you create a molecule or an antibody, and then you have a whole process of humanizing it, like so to avoid such problems. And this is one example for in case of using it in humans. Let's say a protein is a therapeutic, but you can also think of being very efficient in designing enzyme that will just eat away not everything, but like it could be, I mean, technically you would need to have large amounts, but imagine, now I'm going sci-fi, that you have a system, enzyme that is degrading something, that it's self-propagating in a polyreactive, like in a chain reaction. Exactly, viral prions. Like these are all cases of, this could go, you know, into the wrong direction. I don't know if the speed of reactions is big enough to, you know, that you could create it and say, now I destroy the world like it's atomic bomb or something. But they could pose problems of similar scale. Also with, you know, it could be also misused in viral design, viruses that are mutating very efficiently. And then this can be the end of us, because we have seen with Corona that we have issues as a society dealing with pandemics, and these issues have somewhat made us stronger, but have also created a lot of problems in community, not believing in like general immunology. And yeah, these are all, I think, also the problems that we're creating. Similar that with, you know, generative AI and consciousness engineering, maybe. Yeah, I think it's hard not to touch on them, or maybe, yeah. Okay, I will close the recording because have a good, 5 in the chat, 4 new. Okay, that was all about the sound. Never mind. Have a good Easter holiday, everyone, and see you in the next round of lectures with, I think Suvo is... No, next week we have a guest lecture. Yes.
We have to check, but I think that Saptashva is going to be later.
Yes. Oh.